you have to drive 100 miles to get here, then you might reconsider, I don't know. So anyway, we're gonna, on Monday, as you know, we'll talk more about this, but Monday is a holiday again. And next week, we'll do exactly what we did the previous holiday week. And that is, we'll just do lecture stuff. We'll come to lab on Wednesday and pretend like it's lecture. Have a lecture quiz over this tonight. And tonight, we're completely shifting gears and getting into the rest of the quarter. We'll talk about the cardiovascular system. Very germane to healthcare professionals. And this is really the bread and butter class for a lot of you who have career aspirations in healthcare. And plus, it's really cool stuff anyway. And so, is that all I wanted to say? Any questions? Okay, want to make sure you know what's going on, especially this holiday thing really, gets us out of our rhythm. So you want to make sure you know what to expect. I also have another revised syllabus, as if I couldn't do it the first time, with proper dates on it. And my software won't let me get in and see if the dates are correct. But yet, as I look on my flash drive, I see that they are. So I don't see how they could change in between my flash drive and going online. But who knows? And so anyway, check it out. So the cardiovascular system, and basically everybody knows what that does. It consists of three major components. That which is pumped throughout the cardiovascular system. And of course, anytime you pump something, you have to have a bunch of pipes and conduits. And of course, the danger of pumping a liquid medium through a bunch of pipes is what happens if one of the pipes springs a leak. And so we're constantly concerned with that. Plus, you have to have a pump. And in this case, this prodigious pump of ours pumps for our entire lifetime. Pretty amazing. And of course, we all try and take care of it to a greater or lesser degree. And I have all kinds of good advice if anybody wants to know how to take care of your heart. But I think that's one of the reasons Americans are living 10 or 15 years longer, is we're figuring out how to take care of our heart. And for those of us who are a little bit behind on that, there's some really great drugs out there. So one way or the other, if anybody wants any advice, you'll probably wind up getting it whether you like it or not. I used to be an aerobics instructor. Did I ever tell you that? So anyway, I can be a real pain. So anyway, so we're going to talk tonight about blood. Everything you ever wanted to know about blood. Where I was before, they actually had a 2-quarter sequence on hematology where they talked about nothing but blood for 2 quarters. I mean, there's a lot you can say about it. And so it's kind of hard to show a picture of blood, so I just showed you a picture of this heart. Anybody notice anything rather unusual about this heart? Or have we really gotten that far that you would notice? This heart has, from what I can see, has a triple bypass. Here you see a bypass artery there. It's a bypass artery graft. There you see another bypass artery graft. And there you see another bypass artery graft. And presumably, what has happened here is we've got an occlusion in this vessel right here, and we have another occlusion in that vessel, so we bypass the occlusions by pumping straight from the aorta downstream of the occlusion. Otherwise, all the tissue which is downstream from the blockage would die. And when that happens, it's called an infarct. And we'll use the word infarct a lot. It's death of tissue as a result of cutting off the blood supply. It can happen happens in the brain during stroke, happens in the heart. Maybe you've heard of a bowel infarct. It can happen anywhere that a major artery becomes occluded. Okay, so blood consists of two major parts, the liquid part and the solid part, and the liquid part is plasma. Okay, consists of slightly more than half of the volume, and the solid part is all cells. And so a good deal of tonight's discussion will involve getting to know the various cells, and indeed, there are lots of different kinds of cells in blood, all of which are very important, and we'll come to know most of them intimately. Okay, so hematology is the study of blood. Hemo means blood, or hemato. Okay, and so often when they draw blood, not always, but most of the time, they'll spin it down in a centrifuge so that spins real fast so that the heavier stuff sinks to the bottom. Okay, and the heavier stuff is the cells. So what they see when they do that is something that looks like this. All the red blood cells, most of the cellular components are red blood cells, erythrocytes. A minor component of the cells are white blood cells, the leukocytes, and they're slightly lighter than the red blood cells, and the plasma is even lighter yet, although you can tell by looking at it that there's lots of stuff dissolved in it, all kinds of things dissolved in it. It has almost a slightly thick appearance, but of course the solvent is water. Okay. Blood carries fuel, nutrients, and oxygen to all parts of the body and regulates all kinds of stuff and protects us. So let's elaborate on that. Fuel, nutrients, and oxygen also carries waste products from the cells to the lungs, where we dispose of carbon dioxide, and the liver and the kidneys, where various substances we don't want are either biotransformed into things we can use or prepared for elimination by the kidneys and subsequently eliminated by the kidneys. The kidneys can only eliminate polar molecules. Okay. The blood helps in the regulation of body temperature because blood is warm. Okay, so one good index of circulation is just if the surface is warm. And I've heard nurses say that if they walk into the room and the patient is pink, I mean, that's pretty good right there. Have a nice pink patient is a good thing, because that means there's good circulation, and it means also that you have good pulmonary function, because if the lungs aren't working, the patient will be blue. Okay? And blue and cold is not good. Warm and pink is a good thing. And so, okay. Also, we'll see blood is pivotal in maintaining water retention, because much of the water is retained in the blood. pH, which we'll talk all about, and electrolyte, which is just dissolved ions. And electrolyte balance has everything to do with osmosis. So we dredge up the osmosis thing again, as we will for the whole rest of the year. And of course, we have to be able to plug up the pipes, and that's what the term hemostasis means. Literally, it means stopping blood. Okay? And disease prevention, of course, we have cells of the immune system which reside to some degree in blood, or organs which have a lot of blood flow in them. Okay, and so we see the most prevalent cells. It's kind of a toss-up between platelets and erythrocytes, which are red blood cells. okay? All the rest of these are white blood cells, even though they're colored in this picture. But the so-called white blood cells, and so you've all heard about having a high white blood cell count, and typically something in the vicinity of 5,000 leukocytes per cubic millimeter of blood, something like that. 45 to 5,500 is the normal leukocyte count. And if you have less than that, then the computer red flags you. And if you have more than that, you almost certainly have an infection. If you have less than that, you are said to be anemic. And the word anemia is not a good word. Literally, it means lack of blood. That's a very antiquated word, and it's not really a good word to use. Leukocytopenia is a lot better word, and we'll talk a little bit about words like that. And so we'll get to know all these different types of white blood cells. When will we do that? How about now? Okay, so we have erythro means red, cyte means cell. Erythrocytes have one primary duty: they carry oxygen. And they also carry carbon dioxide a little bit. They help process carbon dioxide. 5 million erythrocytes per cubic millimeter. Okay, leukocytes are only a few thousand leukocytes per cubic millimeter. A cubic millimeter is the same as a nanoliter. Thought you might enjoy hearing that. But a cubic millimeter is a little cube, 1 millimeter on each side. You know the smallest hash mark on your ruler? The metric side of your ruler. And so that's a unit that's often used when talking about your blood cell counts. Okay? And so we see these cells have been stained, and so the red blood cells don't look red, but the white blood cells stain purple. And you can see the different kinds of white blood cells actually have different appearances that are really quite easy to identify. There's some platelets, there's a lymphocyte, a monocyte that's involved in mononucleosis. Neutrophil is the most common type of white blood cell. A lymphocyte is a cell of the immune system. There's another one. Looks like the nucleus pretty much fills up the cell, whereas the nucleus, the nuclei have very distinct shapes in these other types of cells, and the shape of the nucleus is how we generally identify them. So people who do work in the labs in hospitals, and we used to have a major in that where I was before, you know, they can easily identify these. That's what you learn to do in hematology. And even I could do it if you gave me a properly stained slide, and I don't even want to learn how to stain them. It's very tedious work, and that's what medical technologists do. People they sometimes call it medical laboratory technology, med lab. And to learn how to do that kind of stuff, learn all about all the different stains that show different things, different ways of culturing bacteria, like blood agar shows up Streptococcus pyogenes that causes strep throat, and things like that. So, okay. All leukocytes come from stem cells which live in the bone marrow, and the stem cells are called hemocytoblasts. Hemo means blood, cyto means cell, blast has a connotation, an embryological connotation, okay? Implying that this is some kind of embryonic organism. So hemocytoblast, if you take it apart, it's pretty easy to deal with. And of course, the hemocytoblast is said to be pluripotent. It has a pattern of gene expression that enables it to become any of a number of different cells. In this case, any of a number of different types of blood cells. hemocytoblasts can't become anything else. They're not totipotent, they are pluripotent. They're within a narrow range, they're capable of differentiating into a number of different kinds of cells. And this is the cells we're going to be talking about. Okay, stem cells, hemocytoblasts are stem cells. And they are pluripotent. Okay. Uncommitted pluripotent stem cells. So, blood cell differentiation, and I know that I have a question on which of the following gives rise to all white blood cells, and one of the choices is hemocytoblasts, and that's the one you pick. Okay. Are you going to pick that next Monday?
Yeah, because we won't
No,
be
you're
here.
going to pick it next Wednesday. Okay. So, let's start by talking about erythrocytes. They are biconcave discs, and they're lacking a nucleus. Okay. The fact that they're lacking a nucleus means they don't live very long. It is the nucleus that maintains a cell for a long period of time. These things are programmed really not to live very long. And there's some pretty good reasons really why it's better to keep recycling them. So they work better. A blood capillary, they're about 7.5 micrometers in diameter. A blood capillary is about 9 micrometers. So these things are forced through blood capillary, single file. Okay? A blood capillary is just a little bit wider than one of these. And maybe you've seen on all the good media, I'm sure you could go to YouTube and find a picture of the heart pumping blood through capillaries, and as the heart beats, you see it move through there in single file. It takes a terrific amount of force to drive a single file column of little teensy cells through thousands of miles of pipeline that we have in our body, because these capillaries, where are capillaries? Everywhere. Yay! So, yeah. So biconcave discs. And all that's inside pretty much is, they're just a little bag of hemoglobin. And this hemoglobin is this gigugous protein molecule consisting of 465 amino acids. Okay? There are actually 4 genes that code for this. And there are 4 heme groups inside each hemoglobin molecule. And this is what a heme group looks like. Really the business end of a heme group is an iron ion. This iron ion can reversibly bind oxygen atoms. It can pick them up and drop them off at the appropriate times. Hemoglobin molecules have great chemistry. They have a whole host of different chemical properties, all of which we'll talk about, and all of which is beneficial to us. They do wonderful things for us. Each iron atom can carry 1 oxygen molecule. Okay? And so a hemoglobin molecule is capable of carrying 4 oxygen molecules. There's just another picture of the same thing. Okay? And when hemoglobin is carrying oxygen, it has an intense red color. When it is not carrying its full complement of oxygen, it has a blue color. So the veins we see on the surface of our skin have a bluish color because they're carrying deoxygenated blood. Arteries are deeper vessels, and they carry blood away from the heart. Okay? The blood has been fully oxygenated. Okay? And the blood goes out and drops off the oxygen to all the cells of the body and comes back, and it's lost a lot of oxygen, and it comes back via the veins, which are the vessels that return oxygen to the heart. And the veins have a bluish color and are close to the surface. And they're blue because they carry deoxygenated blood. See? So. Sickle cell anemia, remember I said there's 465 amino acids? They know what every one of them is, and it turns out that one of them is missubstituted. Let's see which one here. Out of 465, they even have them all numbered. There are actually 4 huge segments, and each one of them has approximately 146 amino acids. And in one of the chains, the chain should have a glutamic acid in it, and sickle cell anemia is a genetic flaw in the gene where the gene codes for one incorrect amino acid. And the amino acid that's incorrectly coded for is nonpolar. This glutamic acid is highly polar. Hemoglobin is dissolved in water. And so on the outside of the hemoglobin molecule, you'd want to have all polar amino acids in contact with the water. So you stick a nonpolar one in there, and the hemoglobin bends around in an attempt to hide the nonpolar amino acid from the water. And in bending around, it loses a whole lot of its ability to function. And it has a totally bizarre shape then. That changes the shape of the red blood cell. And the red blood cell is shaped like a little sickle. Okay? This is a good word to know, especially if you see it in a drug book. Could we prop that door open? Yeah. Push it real hard.
We're good.
Yeah. So, anyway, aplastic anemia is a word you don't want to see next to your name. It means the inability of hemocytoblasts to evolve into blood cells. It's often called bone marrow depression, and radiation is the most common cause of it. It's one of the really bad things that we're talking about, you know, being exposed to plutonium or something like that. Could result in aplastic anemia. There are a few drugs that can cause it, and Tegretol is a very famous seizure medication, and in one out of 300 patients on Tegretol develop aplastic anemia, but there's a big black box in the all the drug books that says aplastic anemia, you know, and they monitor the patients very carefully. Most people don't have a problem with it, but there's some kinds of medications that can cause it as well. Okay, stages of erythrocyte development, and every single type of blood cell goes through various stages in its life cycle, and you know, starting off with a hemocytoblast, and each one has a name. And if you're a medical technologist or work in a hospital lab, you can identify, you can look at that cell and, oh, say, that's a late erythroblast. And that has some significance. For instance, one thing they're very interested in is reticulocytes. If you have an abnormally high reticulocyte count, it probably means you have internal bleeding. And so they look at your retic count, they call it. They suspect internal bleeding, and they can identify all these stages of development in all the different blood cells. Notice that the nucleus comes out at some point. Okay. So how do we know how many erythrocytes to produce? It turns out that the kidneys actually have an endocrine function. The kidneys, because there's a huge amount of blood flow through the kidneys, something like 180 liters a day of blood circulates through the kidneys. The kidneys have been selected to function as the erythrocyte production controllers, and they have detectors sensors that can sense the oxygen content of the blood. And if the oxygen content of the blood is not high enough, then the kidneys sense that and release a hormone called erythropoietin. For people that are anemic, one of the cures is to give erythropoietin. There are various drug names for it, I forget what some of them are, but it's just the hormone, which is very expensive. And so the kidneys release erythropoietin, which tells the bone marrow stem cells to produce more erythrocytes, and so you produce more of them, and the oxygen levels rise. Okay, so if you move to Colorado from here, which is a mile high, what'll happen is there's not enough oxygen in the air, and for the first little while, you'll be out of breath if you walk up to anything. It's really depressing to be a long distance runner and move to a high altitude place and for a while. But what happens is, of course, you don't have as much oxygen in your blood. And so the kidneys sense that, produce more erythropoietin, and after a while you produce more blood cells. And then you can run as much as you used to be able to do before. What's really fun is to live in a high altitude climate and go run in a place like here at Eugene, because there's so much oxygen in the air, you just never seem to run out. So it works both ways. So negative feedback loop in every sense of the word. Erythropoietin causes erythrocyte production, which raises your oxygen levels back to normal. And that's called erythropoiesis. So red blood cell production by bone marrow, stimulated by erythropoietin, which is the hormone. The kidney sense lowering oxygen levels and release erythropoietin. Negative feedback. And typical control system, we even use the little teeter-totter that we haven't seen for a while. Okay? Because erythrocytes don't have a nucleus, they don't live very long, about 4 months, which is pretty short for a cell in the body. And so after a while, they start to die and migrate to the spleen. And the spleen is where they go to die. And one of the duties of the spleen is to break down dying red blood cells. And we recycle the iron, but oddly enough, we don't recycle the hemoglobin. We break it down in a very awkward, cumbersome process because it produces gobs of a very non-polar, bright yellow substance called bilirubin. And because it's non-polar, the kidneys can't eliminate it. And there is an enzyme in the liver that attaches a polar molecule to bilirubin, which allows the kidneys to be able to eliminate it. If the liver isn't happy, it can't do that as much, and the person can't eliminate bilirubin and starts to turn yellow because he's not eliminating bilirubin. Premature babies oftentimes don't have enough of this enzyme and are jaundiced for a while. That's the term jaundice. It just means bilirubin buildup. It accumulates in the subcutaneous fat because it's nonpolar, and you turn yellow. It's hard to get rid of. And we're constantly plagued with getting rid of this bilirubin. It binds to plasma proteins, and that helps us solubilize it in the blood a little bit. But we attach this polar molecule to it, which allows us to eliminate it eventually. Constantly trying to get rid of that stuff, and we're constantly breaking down red blood cells. Okay? And so we break down the hemoglobin and save the amino acids, and we save the iron, okay? And it's bound to a protein called transferrin, which is then used in the synthesis of new hemoglobin molecules. So as the bone marrow builds new erythrocytes, it incorporates new iron, and we pretty much recycle the iron. Although you lose a little bit of it, so that's why we have an iron requirement. But the organic part of the molecule, this bilirubin, we're really plagued by it. Some guys from the Berkeley Chemistry Department discovered, actually bilirubin has a bunch of polar groups in it, but it's shaped like a horseshoe, and the polar groups attract each other and inside the horseshoe, and so the outside of the horseshoe is non-polar. These guys discovered, about the time I was in graduate school, they discovered that if you irradiated bilirubin with blue light, that it would open the horseshoe up and expose all those polar groups, and your kidneys could eliminate it. So they irradiate babies with light, and that helps solve the problems now if they have that problem. And these guys were just discovering that, and I saw the seminar. These guys used to come around every Friday afternoon and give these seminars real late. They did that on purpose to keep us from leaving too early and make us watch these seminars, but I saw some pretty interesting people come through. So after 4 months, the erythrocytes migrate to the spleen and are broken down. Okay, and so that was erythrocytes. Platelets make up a huge part of blood, and we'll talk about them a little bit, but now let's talk about leukocytes. Okay, and there are two general types of leukocytes, granulocytes and agranulocytes. There are 3 types of granulocytes, and if you stain them, it shows up these granules, which are little vesicles, little bags of chemicals that they contain. If they get mad, they'll go through a process called degranulation and release the contents of the granules, almost all of which are highly biologically active and cause some really profound thing to happen. Generally, the profound thing is not good for somebody, either us or some foreign invader. And so the granulocytes contain these chemicals. Agranulocytes don't. Okay, so let's look at those. Possess granules of potent chemical agents which can be secreted through degranulation. Neutrophils comprise 70% of all leukocytes. And the thing you should know about neutrophils, that they're the first cell to the site of an infection. So if you get cut, okay, you always become infected because there are bacteria everywhere, and their neutrophils rapidly migrate to